So carbohydrates, right? The carbs, the sugars, the saccharides. We've been talking about all of this. But if you meet somebody on the street and you ask them about carbs, what they're going to think of is something like this, right? The carbohydrates, the high carb diet, and all that stuff. So if you look at something like this, why do we call these carbohydrates, right? What are the type of sugars that are in a banana, for example? What about a potato? Um, why do we call some carbohydrates good carbs and bad carbs? These are the kind of things that I want us to understand by the end of this chapter, right? And we've been slowly but surely talking about this, but I want to talk about one last thing that will kind of complete the puzzle, and this is the polysaccharides. So let's talk a little bit about what we've been looking at. We looked at individual sugars. We looked at individual sugars such as glucose and fructose, and we saw that these sugars uh, can be ketoses and aldoses. And furthermore, we looked at how we can and we do make circular versions of these sugars, right? So glucose becomes this glucopyranols, and then fructose becomes this five-membered ring. So this is how it looks like. And these, remember, we call them monosaccharides, or just one sugar. But we go one step further and combine one or two sugars to make what we call oligosaccharides, right? So this is a combination of fructose and glucose to make sucrose, right? And this is the sugar that we all love, that tastes sweet. This is sucrose. Now, these, we call them specifically disaccharides, right? And we've looked at a few of these, and these are still simple type of sugars, right? So this is, for example, your sugar that you'll find in your Coca-Cola, or the brown sugar, and so on and so forth. They will be mono or disaccharides, right? But that's not what you find in a potato, for example. There, you'll find polysaccharides, right? What are polysaccharides? The polysaccharides are, well, what the name suggests, right? They are polymers of saccharides, or many saccharides. That's what I think when I hear poly. I hear many, right? And these are chains of saccharides, thousands and thousands. So it's no longer just two or three in oligosaccharides. So when we look at these, and we see these in nature, in humans and in plants, and they usually serve two or three main roles, but mainly two: storage and structure, right? They serve storage and structure, and by storage, it's mainly energy storage. But some of them also serve as recognition molecules, and we'll talk more about that. When you're looking at these saccharides, they can be a homopolysaccharide or a heteropolysaccharide, and this is determined by the individual segments in this polymer. If these segments are similar, let's say a chain of just bunch of glucose molecules, these are homopolysaccharides, right? But if these individual monosaccharides in this polymer are different, let's say a chain of fructose, glucose, fructose, glucose, fructose, glucose, and it keeps going, this is a heteropolysaccharides, right? We're familiar with homo and hetero, and we saw this when we were talking about polypeptides. Alright, so let's look at examples of some of this. So when we talk about storage molecules, we have starch and glycogen. We'll talk more about these, but one thing that you will notice very quick is that starch is a storage system in plants, whereas glycogen is a storage system in animals. Chitin and cellulose are structural molecules that serve structure, and we'll talk more about that. And cell surface polysaccharides are recognition molecules, and these ones are kind of special because they are these polysaccharides, these chains of a sugar on top of a cell, and these work as recognition molecules that enzymes and other signal molecules can come and recognize on cells, which is really cool. So let's start with starch. Right? Starch can have two forms, amylose and amylopectin. By the way, starch is made of glucose, right? It's just a chain of glucose, a bunch of glucose. But there can be amylose and amylopectin depending on what we have. So what's the difference? The main difference is the way we connect these glucose molecules, right? So amylose will have alpha 1, 2, 4 linkages, right? So that means that carbon 1, alpha carbon 1 of one glucose will bind to carbon 4 of the other glucose, right? And then on the other side, the carbon 1 binds to carbon 4, and so on and so forth. In amylopectin, we have branches, and we have branches every 12 to 30 residues, and this number is important, and I'll tell you why later, right? Most of the starch will be amylopectin, so 10 to 30% is amylose, 70 to 90% is amylopectin. All right, so this is how it looks like. Amylose, right? If you look, these are glucose molecules, right? And this right here, if you can see where I'm pointing, this is carbon 1 of one glucose molecule. This one is carbon 4 of the other glucose molecule, right? So 1, 2, 3, 4, carbon 1 to 4. So it's alpha 1 to 4 linkage. It's the same linkage here, same linkage here, same linkage here, and so on and so forth, right? What about amylopectin? Amylopectin, we have branches that are 1 to 6, right? So you have branches that have carbon 1 to carbon 6, right? So actually the normal linkages are the same, right? The normal linkages are 1 to 4 for amylopectin and amylose, and then you have branches that are alpha 1 to 6, all right? So next time if somebody asks you like what type of sugars do we have in potatoes, for example, you'll know what to tell them because you know what starch is made of, right? And you know the kind of linkages and the kind of bonds and the kind of carbons that are connected when it comes to starch. And again, it can be amylose or amylopectin, right? So those are the structures that we're going to be looking at when we say starch or when we say carbs, right? But we're not done. Let's talk about amylose, right? A little bit more about amylose, right? Amylose is purely soluble in water, right? So you guys know about micelles, right? So it forms micellar suspensions or micelles in water, right? And it actually makes, I think I have a picture, a figure that I'm going to show you in a minute, but it forms these helical structures that are pretty intricate, right? So this, so it forms helical structures like these. And one thing to say is, one thing to mention is the iodine fits very well in these helical structures of amylose to form, to produce a blue color, right? And this is important because one thing that we're going to learn in the lab is to test whether something has starch or not. And this, we use iodine. And if something has starch, it turns blue. If it doesn't, it stays brown, because iodine is usually brown. All right. So you might ask yourself, why do we need branching in starch? Right? This is a good question that I like to ask my students. Like, why do we even need branching? And the biggest reason is that we providing access to more molecules of glucose. Right? We'll talk about how, when we're talking about glycolysis and all these other ways to break down the energy, we're gonna need glucose molecules. We're gonna need free glucose molecules to break down, right? So if you have just long chains of glucose molecules, you only have the ends available. But if you have more branches, that means that now you have more ends of glucose available. So that's the main reason why we need branching in starch, right? So this example is whenever you have, so we have this enzyme, right? It's called phospholase, right? It's gonna come in and break down one glucose molecule, right? And we need glucose 1-phosphate to start to use energy. And to do that, we need the ends of glucose. And if we have more branches, we have more ends. That's pretty much the main reason why. All right. So let's go to glycogen. Glycogen is a way to store glucose in animals, right? So check this out. Glycogen is just like amylopectin. So glycogen is kind of like starch, because remember amylopectin is in starch, right? It's just like starch in terms of the connections, right? So it's an alpha 1,4 connections between the glucose molecules, but also it's alpha 1,6 connections between the branches. So it's the same structure. The only difference, there are many differences, but the main difference is that instead of having 20 to 30 branches, instead of having branches every 20 to 30 residues, now we have 8 to 12 residues before you have a branch. So that means that we have more branches, right? We have more branches in glycogen. That's the only difference between glycogen and amylopectin, right? And just like amylopectin, when you add glycogen to iodine, it turns kind of a bluish color, right? So this is an example of glycogen with alpha 1,4 linkages here and then alpha 1,6 linkage on branches, right? And as you can imagine, since we have more branches, glycogen is even more available to be broken down, right? It's even more available because it has more branches. It has more free glucose ends to be used, right? So let's talk about structural polysaccharides. So structural polysaccharides are similar. They're similar to storage polysaccharides, right? But there are small differences that make them serve as structure rather than storage. For example, cellulose which we find in plants, has better 1,4 linkages. Emphasis on beta, because starch and glycogen are on alpha, right? So beta 1,4 linkages will look different. Look at this. We're using the beta carbon, the beta carbon instead of oxygen, and remember that all it means is that the oxygen is down here, right? The oxygen is down here instead of being up, right? So here the oxygen is down, here the oxygen is up, right? So one is alpha, the other one is beta. And you can see, look at how the fact that oxygen is down versus oxygen being up makes it look different in terms of structure, just because one is alpha, the other one is beta. And if we can go, I think I have a figure where we have where it polymerizes, right? When they layer together like this, then it's going to layer differently, whether it's alpha or beta. So this is how cellulose serve as a structural polymer. Right? This is how it looks like. So cellulose is mostly found in the cell walls of pretty much all the plants, right? So wood and bark of trees will be made of highly organized structures. So cellulose is very important because we use it in cotton, right? Cotton is made of cellulose. So when you see cotton, now you know that it's just going to be beta 1,4 linkages of cellulose. Right? There are so many cellulose derivatives in the industry that are used as structural molecules, namely in some forms of plastics, some forms of clothes, right? They use cellulose. Cotton acetate is one of them, where they just add an acetyl group to cellulose. Really cool. So cellulose, one thing to note is that we cannot digest cellulose. We break it down to something else, but guess what? Animals can digest cellulose. That's why animals can eat grass and we cannot. That's because we cannot digest cellulose. What's the main reason why animals can digest cellulose and we cannot? Is because they have a bacterial cellulase. It's an enzyme that breaks down cellulose in the rumen. Right? I don't know if you guys learned the digestion of these. We call them ruminants. And they have like compartments. They have intestines that are made of compartments, and it's pretty intricate. And in one of these compartments, that's where we have these bacteria that have cellulase, and this cellulase breaks down cellulose that they take in. All right, so chitin is another one, and this is found in the skeletons of insects, like spiders, and some fungi, and it also serves as structure, right? And this one, speaking on structure, it's kind of like cellulose, except on carbon 2 you have an acetyl group, right? An N-acetyl group. So look at this. Look at the bottom here, right? You see this? So cellulose usually just has OH at the bottom on carbon 2, right? But chitin, you have an acetyl group. That's it. So this is a glucosamine. This is a glucosamine. So it's an N-acetyl glucosamine. uh, one unit, N-acetylglucosamine. So instead of just being a glucose unit, it's an N-acetylglucosamine, and then you have a bunch of units of this. That's chitin, and as you can see, it's also using beta, right? Beta 1,4, just like the other one, right? So that's the only that's the difference number one. Difference number two is whenever cellulose get together as strands, like I showed you in that other picture, like this one, right? They are parallel, right? They are parallel in conformation, but chitins can be parallel and anti-parallel, and this comes from this acetylamine group here, right? So what about other structural molecules? Let's see if we can look at other ones here. Just give me a second. Agarose. Agarose is another one of my favorite because I use agarose in the lab a lot. Agarose is a polymer of galactose. Agarose and agaropeptin is a polymer of galactose. And then glycosaminoglycans, right? These are very interesting, and we'll talk more about those. But just look at the name, right? Glycosaminoglycans. So it's a glucose that has amine in it. So it's an amino sugar, right? And these ones are pretty interesting, and we'll talk a little bit more about them. So agarose is very interesting because it forms a helical structure, right? We talked about how it's made of galactose units, but these galactose units form a helix like this, right? And when it forms a helix like this, then these intricate structures can be used in the lab as a gel. So we use agarose gels to separate DNA, for example, right? Or something that I recently did was to use an agarose column to separate different molecules of different sizes, right? Because these intricate agarose structures can let things through, right? It's very, very cool, and we use agarose a lot, especially if you're doing DNA work. Glycosaminoglycans is just like the one that we looked at. It's a glucose, right? And then it's usually two molecules, right? One is just a sugar, the other one is a sugar bound to an amino group. So glycose is one side, and then aminoglycan is the other. So glycose, aminoglycan. That's where they come from. You don't need to memorize these, but you need to be able to recognize them and know what they are. Glycosaminoglycans are used in many different places. One that I can think of is heparin. Heparin is also an important molecule that we use. There's one of my collaborators in research is using heparin to actually work on wound healing and things like that. But heparin is a natural anticoagulant, and it's very widely used in research. You can also find these in the eye, in the connective tissue, in the cartilage. You can find these in the skin, in many other places. Another one that we talk about is proteoglycans, right? And these ones, as you can imagine, look at the name, proteoglycans, right? You hear protein and then you hear glucose, and we'll talk about these. And these ones are, aminoglycans, glycosaminoglycans are found in proteoglycans as well. So let's talk about bacterial cell walls. And the reason why we talk about bacterial cell walls in this chapter is because bacterial cell walls are made of sugars, right? Among other things, they're made of sugars, and specifically peptidoglycan. And what do you think when you hear peptidoglycans? A peptide group and a sugar group, right? Peptidoglycan. So if you look at this figure down here, you have gram-negative and gram-positive bacteria. If you ever heard of bacteria, you heard of these. And we have really cool microscopic test of staining bacteria to see if they are gram-positive or gram-negative. But when we're talking about gram-negative or gram-positive bacteria, we're talking about their cell walls. Gram-negative bacteria have a lipid bilayer separated by a peptidoglycan layer. These are gram-negative. Gram-positive have a thick peptidoglycan layer on top of their one lipid bilayer. Right? You see the difference? That's a big difference. And this is what manifests in pretty much what the bacteria end up doing to your body. So if we kind of zoom in, what we mean by peptidoglycan, it's a peptide, right? So look at these. These are different amino acids, right? This is a peptide. This is alanine, lysine, glutamate, and then alanine as well. So it's what we call a tetrapeptide, right? Which 4 amino acids. So this is a peptide, right? And it's bound to a sugar. Look at this, right? This is a sugar, right? But it's a sugar that's also bound to an amino group, right? So that's what we mean by peptidoglycan. Peptidoglycan, right? And if we zoom out, this is usually how it looks like. It's the sugars, right? And the sugars are glucosamine, right? Acetylglucosamine. And then they are bound together by these tetrapeptides, right? The tetrapeptides. And these green ones is what we call a pentaglycine crosslink. So this is also a pentaglycine. So it's also a peptide or 5 glycines that are bound to it, right? So this is in the gram-positive. So going back to this gram-positive, right? So we're zooming into this layer to see how it looks like, right? It's a layer that's composed of sugars and sugars that are bound by peptides, 4 peptides, and then a pentaglycine crosslink, right? This is what it's made of, gram-positive. In the gram-negative, we don't have the pentaglycine crosslink. We don't have the crosslink, right? And that's what makes them be just one layer in gram-negative and a bunch of layers in gram-positive, because here we don't have the pentaglycine crosslink, right? And I think that's enough. That's enough on bacteria, but this is another example of how they look like. But that's what differentiates positive and negative bacteria, right? Another thing that you'll see is there's something we call Lipo Polysaccharides, right? And this happens on the outside of bacteria. So on the outside of bacteria, you will see some of these chains that you see right here. We call them Lipo Polysaccharide. And think about what that name means. Lipo Polysaccharides, right? It's like lipid bound to a polysaccharide, right? And this is how they look like, right? You have a lipid group, right? Lipid group that's anchored in the membrane, right? And then you have a polysaccharide. Let me see if I can do it. So the lipid is this one down here, and we'll talk about lipids in the next chapter, but lipids are down here, and then you have a polysaccharide, and up here lipopolysaccharide, right? And these are really important, and they work as signal molecules, and they can also do other roles as well. So animal cells also have polysaccharides on the outside, right? And they can be those lipopolysaccharides, but sometimes they're just what we call glycoproteins. So one of the things that I like to ask is, what's the difference between a glycoprotein and a proteoglycan? And we'll talk more about that, but glycoproteins are what it sounds like. They are proteins that have a sugar molecule bound to them, glycoproteins, right? So this is an example. Look at this. You have a serine group right here. So think of this serine. It's not just serine. It's a long chain of a protein. But on serine here, we come in, we attach a disaccharide here, right? So this is a glycoprotein. And we have two types of glycoproteins. One that's bound, we call it O-linked because, well, it's linked by an O group. And then we have ones that we call N-linked. N-linked are bound by the N group, right? So here, for example, it's bound to asparagine. So asparagine, the N of asparagine binds to this sugar molecule. But here, it's a serine group, and then the O of serine binds to the sugar molecule. So serine can form an O-linked saccharide, and then asparagine can form an N-linked saccharide. So those are the two types of your glycoproteins. Proteoglycans are special types of glycoproteins. How special are they? The sugar molecule that's bound to these proteins will be glycosaminoglycans, right? So it's a bunch of terminology that you need to know, but glycosaminoglycans. Remember the glycosaminoglycans? We talked about them. It's these molecules that have a glucose, right? Not a glucose, but they have a sugar molecule. Let me see if I have an example of aminoglycans. They have a sugar molecule, and then we'll find them eventually. There they are. You have a sugar molecule bound to another sugar molecule bound to an amine, right? So glycosaminoglycan. Glycosaminoglycan. That's what we call glycosaminoglycan, right? And then if you take glycosaminoglycan and you add it to a protein, we make proteoglycans, right? So these are glycoproteins. There are special types of glycoproteins called glycosaminoglycans. And these are important because you will find these on the outside of the cell a lot, right? If you look at your animal cell, they will be around, and they form what we call a glycocalyx, which is kind of a sugar layer around the cell. So you've heard of proteins around the cell, but you have a lot of sugar molecules around the cell, and these are made of proteoglycans. And these can modulate cell growth processes, cushioning of the joints, and they can also serve as signal molecules and so on and so forth. So this is an example of how they look like, right? So this is an example of, well, a proteoglycan, right? You have the protein chain up here, and then it's bound to the glycosaminoglycan, right? And some can be O-linked, and some can be N-linked, like we saw, right? So I think that's enough. That's enough of polysaccharides that we talk about. One thing that I'll talk about is, as you can imagine, we talked about how these are found around the cell, and depending on what type of sugar molecules that you have around the cell is going to give off a different signal, and it's going to, have different things that enter the cell depending on these. And a network of these sugars is what constitutes the glycome. The glycome is a complete set of glycans, right, and glycoconjugates that are made by cell or an organism, right? So there's a big study that's called glycomics. So when I say glycomics, you have genome and genomics. So you have glycome and glycomics, and scientists in the glycomics have gone as far as making a sugar code, just like a genetic code, right, to show how things are, made and how signals are transferred in terms of sugar molecules. So the sugar molecules are not just structure and storage molecules, but they can also be signal molecules um, that can form a network that we call the glycome. Right? So I think that's enough about the polysaccharides, but by now, if somebody asks you about um, polysaccharides, you know what to tell them. One thing that I'll talk about last is lectins. Lectins is a family of proteins that bind carbohydrates with high specificity and high affinity. And whenever they bind to these, this is how I was talking about how we transfer the signals and how we recognize specific cells that have specific sugars. These are the proteins that are responsible for that. We call them lectins, and lectins is not just one protein, it's a family of different proteins. But I won't have specific lectins because I don't think we're going as far as that. But I think that's enough. That's enough for carbohydrates. Mm, by now, you should be able to tell in any type of food that you have what type of sugar that you have in there, and you should be able to even draw it and tell me what type of linkages you have, and what carbon number that linkage is at. I think by now we know how to do that, but that's it. I'll see you next time.